Hey everyone, and welcome. So today, we're going to pop the cap on something you've probably all got sitting in a medicine cabinet somewhere, hydrogen peroxide. I mean, it seems so simple, right? But its slow, silent breakdown is actually hiding some really fascinating chemical secrets. Okay, let's dive into this and figure out what is really going on inside that little brown bottle. So, right off the bat, here's the central mystery. We all know hydrogen peroxide is unstable. It's basically always trying to break apart. And yet, you can buy a bottle and it'll sit on a shelf for a very, very long time without much happening at all. You know, it's like a reaction that's always on, but the volume is turned way, way down. And that really begs the question, doesn't it? If this stuff is always decomposing, why isn't it more obvious? Why doesn't the bottle just empty itself out? Well, this is the case we're going to solve today. We're going to investigate not only why it's so slow on its own, but also what it takes to make it happen incredibly fast. Now, on paper, the chemistry looks pretty straightforward. You've got two molecules of hydrogen peroxide, and they turn into two molecules of plain old water and one molecule of oxygen gas. That's it. So the ingredients are simple, the products are simple, but the real story, the juicy part, is all about the speed of that transformation. So if the natural process is just a slow crawl, how do we get it to break into a full-on sprint? Well, we need a spark, something to kickstart the whole thing and get it moving. And in chemistry, that spark has a name. It's called a catalyst. So what exactly is a catalyst? Honestly, the best way to think about it is like a matchmaker at a party. It goes around, finds molecules that should react, helps them get together. But once the reaction happens, the matchmaker just steps back, totally unchanged, ready to do it all over again. It makes things happen, but it isn't part of the final result. For our little investigation today, our chemical matchmaker, you could say, key suspect, is a substance called potassium iodide, or just Ki for short. We're going to use this stuff to dramatically speed up the decomposition of our hydrogen peroxide. Okay, but this brings up a pretty practical problem. I mean, when you mix these two things, you just get a bunch of fizzing oxygen bubbles. How in the world do you turn fizzing into actual hard data? How can you precisely measure the speed of the reaction? The solution is actually pretty clever. What you do is you seal the whole reaction up in a container that has a pressure sensor. See, every single bubble of oxygen gas that gets produced will make the pressure inside go up. So, by measuring how quickly that pressure rises, we get a direct measurement of how fast the reaction is going. More pressure means more oxygen, and that means a faster reaction. Simple as that. So the core of our experiment really boils down to just these three steps. First, we mix our hydrogen peroxide and our potassium iodide catalyst in a test tube. Then, we immediately seal it up with that pressure sensor. And finally, we just sit back, watch, and record how the pressure climbs for 5 minutes. Now, of course, like any good detective, we can't just run the experiment once and call it a day. To really solve this case, we've got to gather clues. And we're going to do that by systematically changing the conditions, just one variable at a time, to see what really affects the speed. All right, here's our whole investigation plan laid out. Think of part 1 as our baseline, our control group, 3% peroxide, a specific concentration of our catalyst, all at room temperature. Now look at the other parts. See how each one just changes one little thing? In part 2, we cut the amount of catalyst in half. In part 3, we dilute the peroxide. And in part 4, we turn up the heat. So our very first clue is going to come from comparing part I and part 2. The only thing that's different here is the amount of our catalyst, the potassium iodide. So will having fewer of those matchmakers at the party slow down how fast our peroxide molecules react? Let's see. Next up, we're going to look at the main ingredient itself, the hydrogen peroxide. For part 3, we're using peroxide that's only half as strong. So the question is pretty simple. If there are just plain fewer peroxide molecules floating around, does that slow the whole thing down? And for our third and final clue, we're going to investigate temperature. We're running the exact same reaction as our baseline, but this time it's in a warmer water bath. Does giving all those molecules a little more energy by heating them up actually make a difference in how fast they react? Okay, so what did we find out? This is where it gets really interesting. This chart shows the initial speed for those first 3 experiments. You can clearly see that part I, our baseline, is the fastest by far. When we cut the catalyst in part 2, or the peroxide in part 3, the rate just tanks. It drops significantly, and that tells us something huge. The concentration of both of these substances is absolutely critical to the speed.
All right, we've gathered our clues. We know that concentration matters. A lot. And we know that temperature plays a role too. So now it's time for the big reveal. Let's use this data to decode this reaction's deepest secrets.
What's so cool is that all our data lets us write something called a rate law. And this is awesome. It's basically the reaction's unique speed recipe. It's a mathematical formula that can tell us exactly how changing the concentration of the peroxide or the catalyst will change the overall reaction rate. It's like having the reaction's secret code.
And what about that temperature clue? Well, that helps us calculate something called the activation energy. You can think of this like an energy hump or a hill. It's the minimum amount of energy the molecules need to have to actually react when they bump into each other. A catalyst works by basically finding a shortcut, a path with a much smaller hump. And our temperature data lets us calculate just how big that hump is. But here it is, the biggest reveal of all. All the clues we've gathered, they all point to a major plot twist. That simple one-line equation we looked at way back at the beginning, yep, that's not the whole story, not even close. The reaction is actually way more complex, and it happens in multiple steps.
And this right here brilliantly shows what the catalyst is really doing. So here's a proposed two-step mechanism. In step 1, the peroxide reacts with that iodine ion from our catalyst. But then look at step 2. A second peroxide molecule reacts, and in the process, it spits that original iodine ion right back out again. This is why a catalyst speeds things up without getting used up. It gets involved, for sure, but it always comes back out totally unchanged at the end.
And this leaves us with one last really fascinating question. A multi-step reaction is kind of like a relay race. The whole team can only go as fast as its slowest runner. So looking at this two-step process, our data can actually help us figure out which of these two steps is the slow one, the one that's acting as the bottleneck, and setting the true speed limit for the entire decomposition. But figuring that out, well, that's a puzzle for another day.